So I'm going to talk today about the natural products from marine invertebrates in Antarctica. These studies have been done in the frame of several grants, Actikim, Ecokim, and some others over the years. And so in our lab, we work in different research lines, um, most of them related to natural products, but others not so much. And we are looking at how these compounds are changing with different environmental conditions, the variability, the production, and also the bioactivity. Um, we are also looking at how the production may be affected by stress situations. And for the bioactivity, which is what I will explain a little bit more today, how can we use computer-aided drug design to help us in finding potential bioactivities um, in our compounds? Also, we do some other research studying the microbiome of selected invertebrates, and we are analyzing human impacts and the effects of climate change. This is related to the variability of the natural products because we are doing experiments in changing the temperature and observing how the compounds in these organisms may be different. So in our Blue Bio project that just finished now this June 2020, we've been looking at this variability, production, and bioactivity. And we're comparing some related species in Antarctica, Mediterranean, and the tropics. I'm going to go very quick over the variability and production, and I will talk more about the bioactivity today. So for the variability, we did some temperature experiments by increasing the temperature and keeping some animals in warmer conditions. And we compared this in Mediterranean, Antarctic, and Tropic. I'm showing you here some results, preliminary results of the bryozoans. We used the Antarctic species Imantozoo antarcticum and the Mediterranean Chartella tenella, and we use 5 replicates. We use controls that were frozen just when they were collected, and 3 different temperatures. The first one was quite similar to the field temperature, and the other 2 were like 5 degrees increase in each case. So this is the work of a master's student in my group, Silvia Borrull, and what she has seen is that the amount of compounds decreases with temperature, but also the diversity of the compounds, more or less in a similar way in the Mediterranean, which is in the top, and the Antarctic, which is in the bottom. And the first temperature, which is more similar to the real conditions in the field, it's more or less the same in the Mediterranean, but it decreases a little bit in the Antarctic species. We are finding similar results in mollusks, in cnidarians, in sponges, but we are still analyzing some of the data. So these are just a preliminary taste of our results, but in fact we can see that yes, the increase in temperature affects the production of the compounds, which means if these compounds are used for defense, means the animals will potentially become unprotected against predators. Another species we've been looking at is the Sislac Doris kerkelenensis. We've been working with this animal for quite a while now. This species is able to build its own compounds for defense, and this may be highly variable in the basic skeleton. They are titerpene diacylglycerols, but they may be a little bit different depending on the population and on the individuals. So if we collect animals from different parts in Antarctica, as also some other researchers have been doing, especially Bill Baker's group, then we can see that specimens, each column here is one specimen, okay, from the Weddell Sea or from the Ross Sea. You can see that some specimens have different molecules, but not only different molecules, they are completely different skeletons. And so this is kind of amazing. There is a work by Wilson and co-workers that describe this could be cryptic species, which could explain part of the variability, but still doesn't explain why in the same population we have individuals so different. So anyway, we are looking at this, how do they build these compounds with these different skeletons. Probably there is enzymatic arsenal here that has a role in this the formation of these compounds. And what we know is that the small individuals, when they are in the eggs, they don't have these compounds. When they are 2-3 millimeters long, they don't have these compounds, and the compounds are not in the prey sponges they feed upon. So they build these compounds at some point. There is something that triggers the production of these compounds. Also, we think that they may be affected by temperature. We've run the same experiments that I show you with the bryozoan, with this species, and we are analyzing the data now. But also we've run some other experiments by stressing the animals, trying to figure out if this pattern of production of natural products changes or not with this stress. So we have the experiments done with freezing the animals directly in liquid nitrogen after collection, then doing some stress and after the stress. So work is in progress, and hopefully soon we will have the results to explain you what happened with these animals. So the third part of my talk is about the bioactivity. It's very difficult when you have a compound to figure out whether this compound could be useful for something or not. Can it be useful for anti-fouling or for anti-inflammatory or against cancer, or how do you know? So you sometimes you have enough amounts of compound to do different assays, but sometimes you don't, and then you have to choose which assay do you do, because you have not enough to do a wide panel of tests to see, the potential applications of these compounds. So Laura, my student that did her PhD, she presented it just 6 months ago, was looking at this computer-aided drug design and discovery with our molecules, and she tried to use in silico studies to find these potential therapeutic indications, and she applied this especially to the most potentially active molecules that we found. Here is Meridianins. Meridianins are from a tunicate. We collected in the Weddell Sea, but also in the Antarctic Peninsula in many occasions, and we've been working on the biological activities. So we were interested in finding potential applications too, and she found, I will show you now, she found it could be potentially useful for Alzheimer's disease. Also some related compounds, because they were structurally similar, could have similar role. Also some derivatives of meridianins, and these compounds were in fact inhibiting a particular kinase protein, GCK3 beta, and inducing structural synaptic plasticity in cultural neurons. So we were able to combine this computational work with experimental work. I'll explain you how did we do that. So the advantage of using this computer-aided drug design is that you don't need any sample, you just need the structure, and then you need to know how to run the programs in the computer, which is the most difficult part, let me tell you. It really allows you to predict the possible effects and side effects and improve viability and everything, but just in the computer, okay? And you can do some screening, reducing the number of molecules and limiting the number of experiments and optimizing the use of your time and money. So what we did was start with a bunch of about 100 molecules we selected, and try to identify possible targets. You can do this two ways. One is looking at the structure of your molecule, and the other one is looking at what your molecule binds to. And so we did both and combined them, and then you get to screen the most powerful drug candidates in your list of compounds. So we, after doing a first virtual profiling validation, we got like 75 complexes, and at the end we arrived to 11 complexes that were related to 6 molecules. um, these molecules were active at several kind of potential diseases like neurodegenerative, cardiovascular, and others. And we were very interested in knowing what was the possible applications of these compounds. So Laura did a lot of docking studies, molecular docking and others, and virtual profiling to find possible targets related to these neurodegenerative diseases, and also the toxicity, because of course we thought meridianins, they are very potent alkaloids, so probably they are toxic. But I will tell you now. We found that meridianins were potentially very active against many diseases, as I said before, especially neurodegenerative. And then when we look at the inside this neurodegenerative box, what were they more potentially active for? It was Alzheimer's disease. Okay, this potential activity, we wanted to test it with more detail. Here you have the structures of all these molecules found in these organisms, these tunicates, Oplidium falglaticum and Oplidium mediterraneum. Sorry, not Mediterraneum, Meridianum. Okay, and of course this was very relevant for us because, as you know, Alzheimer is a very important disease. It has no cure and is degenerative, and so there is nothing you can do if you have this sort of dementia. So really we need to find something, and if we could help with our Antarctic molecules, then that would be great. Okay, so the problem with this disease, the most obvious effects are the accumulation of some senile plaques composed of beta amyloid peptides, and also it's been observed that there is an abnormal phosphorylation of the tau protein. Most treatments are focusing on the second aspect, the beta amyloid peptides, but some others are looking now at the tau protein. That's what our results were taking us to, that our compounds, meridianins, were affecting especially tau protein kinases and dual specificity kinases, and all of them were involved in this potential blocking of the Alzheimer cascade. So this could be a good strategy to reverse the tau phosphorylation and to be used against Alzheimer's disease. So we found these possible targets, and we validated the results through the literature, and we proposed that meridianins could be classified as kinase inhibitors, and we proposed that the presence of the bromine in the molecule could be important for the potency of the molecules. Okay, so the next step was to look at whether these bromines or the skeleton of the molecule were more important, like the indole skeleton and the bromine atoms. So we compared these, and also with some derivatives, and Laura was able to figure out how the molecules we were using impact, contact and bind to the docking areas. And well, I'm not going to enter into the details here, but just to let you know that both things, the indole scaffold and the allogen, are equally important. And actually some compounds, derived compounds, were proposed as very good hits. Okay, so this is all Laura results, and so we went further to see what was the specific effect on GCK3 beta, because this was the responsible molecule for the phosphorylation of the tau protein, in a way that if you are able to inhibit this enzyme, it will decrease the tau hyperphosphorylation, and so it means it will decrease the neurofibrillary tangles. Okay, so we did that, and we found that most meridianins, the meridianins we used, were very active. We also tried other compounds in the bottom, you can see from a mollusk, but I'm not going to talk about this today. And so Laura was able to figure out the binding energies and all the details of this binding, and it was really interesting that also the ATP cavity served as a binding place. So in fact, that was it indicated an allosteric relation. Okay, so we also tried these Meridianins in neuronal cultures of mouse in the lab, and we observed that the molecules were not toxic. They didn't affect the viability of the neurons, but they were really important at, as you can see in the graphs here on the left, at the phosphorylation of the GSK3 beta related with SERID9. I'm not going to enter into all these details, but just remember this point that it's not toxic, it's doing the effect against the GSK3 beta, and additionally we found that they are inductors of structural synaptic plasticity. Okay, both compounds, Meridianins and others, but as I say, I'm only talking about Meridianins. Okay, so as I say, they don't affect neuronal viability. You can see here there is no statistical difference, and so this indicates that Meridianins are able to inhibit the activity of GSK3 beta through an ATP competitive and allosteric mechanism, which means really they could be potential drugs to treat Alzheimer, although of course more research has to be done in this sense. So just to conclude, many new things are happening now. Many new developments are opening completely new perspectives in natural products research of Antarctic marine benthic invertebrates, and many things are yet there to be discovered. So we have to keep working on that. I just want to say thank you for all the funding and the people helping and participating in cruises and research at all levels. And I just want to say that this is a teamwork, and it's a shame we still have political prisoners and exiles in Europe in this 21st century, and I want to dedicate this talk to them. And here I put my address, my email address, so in case you have any doubts, please don't hesitate to get in touch, and I'll try to answer to your questions. Thank you.